The topic for today's session is diagnostic microbiology. Diagnostic microbiology is also called as clinical microbiology. So here the diagnosis means to identify the organisms which are involved in the disease causation. So everything that is studied under this particular thing, that is the different scientific methods that are utilized or adopted to identify the etiological agent that is involved in a disease causation is called as diagnostic microbiology. And here in this particular diagnostic microbiology, we will be studying different steps, how the specimen is collected and how it is transported to the main laboratory where the further tests are usually carried out to identify the organism that is involved in the disease causation. So here first thing that we'll be studying in diagnostic microbiology is the collection and transport of specimen from the microbiological examination. So here the specimen represents a portion of the particular part which is harbored with a microorganism and involved in the disease causation. So specimen is usually collected in aseptical method, and then this is transferred to the microbiological main laboratory where the further processes and procedures are carried out to identify the organism that is involved in the disease causation. So here the collection of clinical specimen, the clinical specimen as already described, it is a portion which is involved of the human body which is usually attacked with the microorganism, and this is a portion that is usually collected to identify the microorganisms. Next thing is the collection of the specimen should be in good condition. Now the quality of the specimen completely represents the result that you get from that particular specimen. So the result is dependent on the type and the specimen or the quality of the specimen that is usually collected to identify the organism. So the specimen that is collected should be from the diseased area. It should not be from any other part until and unless it is a organism that is causing a systemic infection where blood is also involved. Apart from that, specimen should be only from the diseased area but not from the other part of the body. So collection of the specimen should always be in a good condition. That means it should be aseptic in nature without any contamination with the other organism. Otherwise, there will be a problem in the diagnosis of the particular agent. And it is a representative of a disease process. The specimen is the part that usually represents the disease part in our body. And there are certain precautions that have to be taken while collecting the specimen. And it is the work of a clinical microbiologist who usually carries out the different scientific methods to identify the organism from the diseased part. So it is the work of a clinical specimen begins with the collection of the specimen to the identification of organism, which is a final step in a clinical microbiology. So clinical microbiologist has to look into many aspects before collecting the specimen and carrying out various techniques to identify the microorganism. So the precaution that has to be taken up while collecting the specimen is that he should not harm the patient. In case of lumbar puncture or when collecting specimen from the children or from catheterization to collect the urine sample, it should be seen to it that no patient is harmed. And the next is we should ask the patient whether he was already on an antimicrobial therapy or not. That is, if the person is already on an antimicrobial therapy, it will suppress the growth of the pathogenic microorganism. So ultimate goal of the diagnostic procedure is lost when the person is already on an antimicrobial therapy. So here as already described, diagnostic microbiology usually deals with identification of organism that is involved in disease causation. So if the person is already on antimicrobial therapy, then there is the suppress of the growth of the pathogenic microorganism, which ultimately cannot be identified. And next thing is, whatever the containers in which this particular specimen is collected should be completely sterile in nature. That is, it should not be contaminated with other organism, otherwise confusion in the diagnosis will take place. And the quantity of the specimen that we are collecting should be in adequate quantity. That is, it should be seen to it that the quantity that we are collecting is more, because many tests have to be performed in the later. So you cannot call the patient again and again to collect the specimen. So adequate quantity has to be collected before itself so that more tests can be performed on a single specimen. And proper instructions to the patients have to be given when the patient himself is giving his specimen, in case of urine sample or fecal sample or any other sample which the patient is voluntarily coming up to give. So in that case, proper instructions instructions have to be given to the patient for the collection of the specimen. And aseptic collections, because should not be contaminated with the other organism, and suppose we are collecting the specimen from the skin, it should be disinfected so that the commensals are usually eliminated and only the organism that is involved in the disease causation is taken up. And when collecting the fluid specimen, it should be seen to it the container is completely not filled to that brim, otherwise it might lead to contamination. So a little space is left over the container so that there is no contamination of the specimen. And when taking up the specimen with the cotton swabs, like more precautions have to be taken up, because these swabs are exposed to the external environment and they may get contaminated easily. So a modified type of cotton swabs that are made up of calcium arginate or charcoal dipped cotton swabs can be used to prevent the contamination. So different areas of the body which are usually infected by different types of microorganisms. So let us see in detail about the different part of the body that is attacked by different types of microorganisms, and what are the specimens that are collected from this particular areas. First I will start with the skin. So the main purpose of collecting the skin organism is to identify the organism which are usually involved in the skin diseases. So here the best method to collect the specimen is a swab. So again, when collecting the specimen from the skin, it should be in aseptic condition, and no antibiotics or antiseptic should be applied. Only washing with the soap or the water should be done, and no antibiotics, because again it might inhibit the growth of the pathogenic microorganism. And these swabs that are usually used in collecting the specimens are moistened with saline and then dipped in the nutrient broth and pressed gently on the infected area and collected with the specimen. So in case of crust or scabs, you cannot make use of this swab, but directly the part of the crust or the swab will be taken up in a sterile bottle. And in case of viral infections, these specimens can be collected with the help of capillary tube, which are made up of plastic and completely sterile in nature. And for fungal infections, if the hair or the nails are infected with the fungus, then the part of the hairs or nails or scraping from this particular areas can be taken as a specimen. Now coming to the upper respiratory tract, so respiratory tract can be divided into upper and the lower respiratory tract. So each of this part is usually attacked by different types of microorganisms. Usually in the upper respiratory tract, it is the aerobic microorganisms, and people usually suffer from the aerobic type of infections from the organism. So the parts that are usually present in the upper respiratory tract are the oral cavity, anterior nares, and then you have perinasals or pernasals. So coming to the oral cavity, here the specimen is again collected with the swabs, where the ulcerated or the patch lesions are gently rubbed with the swab that is dipped in a saline, and then it is introduced into the particular media where the isolation of organisms will be done. And from the anterior nares, it is again, if there is a pus in that particular area, then it can be collected with the swab, and if no pus is present, then it is moistened with the swab that has been dipped in the saline and then introduced into the culture media. Now coming to the throat, here the person or the patient is asked to open his mouth wide open, and then the tongue is depressed, and then the swab is taken to collect the specimen from the tonsils and the pharyngeal mucosa. Now coming to the nasopharynx, here a special type of pernasal swabs are used to collect the organisms that are involved in the pulmonary diseases such as Bordetella pertussis. So here the pernasal swab is a specially designed swab which is having a wire and bent at one end. The bent end usually has a cotton swab with which you can collect the specimen. So this wire usually inserted through the nasal passage into the nasal cavity, and then the specimen is collected by firmly rubbing onto that particular area wherever the infection is suspected to be there. So now coming to glottis and epiglottis, here again you can make use of swabs which are made up of the cotton swab, and they are usually collected the specimen that is representing the inflamed and the ulcerated area. So in case of children, special care have to be taken place to prevent airways that results due to laryngeal spasms. So now next to the pernasal sinuses, again here the swab can be used, or you can also make use of an aspiration method, to aspirate the contents with the help of a syringe or a needle, where the contents are taken up in the syringe or with the help of a needle. And with the viral infection, it is a fine plastic tube. So these are the methods that are mostly used to collect the specimen in the upper respiratory tract. Now coming to the lower respiratory tract, mostly involving different types of organisms, and the person who suffers from lower respiratory tracts are usually said to be infected with the Mycobacterium tuberculosis. There is suspension of this bronchial infection in the lower respiratory tract. So here we cannot make use of the swabs. We cannot insert it to the lower respiratory tract. So we can, the best specimen that can be used over here is the sputum. So here the person is asked to cough, and the sputum is collected down from the bronchial tree, and it should be collected in aseptic condition. Otherwise, when it gets mixed with the saliva, it might lead to the contamination with the commensals. So here again aseptic conditions have to be followed to collect the specimen of the sputum. And aspiration can also be done from the bronchi or trachea with the help of a flexible fibrio-optic bronchoscope, mostly used for the children or the patients who cannot give the sputum voluntarily. And in that case also you can make use of other specimen that represents the disease area, such as you can go for the aspiration from the nasopharynx, or you can go for the laryngeal swabs or gastric juice. Mostly in case of children who cannot give the sputum voluntarily, you can also go for gastric juice, because it is said that this gastric juice may harbor certain types of microorganisms that is directly coming down from the lower respiratory tracts. So apart from this, initially it was bronchial washing that was used to identify or used as a specimen, but nowadays it is of no use, as because this bronchial washing will contain most of the contaminated microorganisms, that is the normal flora that is present in that particular area. So this is about the lower respiratory tract. So upper respiratory tract and lower respiratory tract. The difference is, in the upper respiratory tract it is easy to isolate the specimen or collect the specimen with the help of a swab, and in the lower respiratory specimen is usually the sputum. Gastrointestinal tract. It is a tract where most of the organism can be present in our human body, and mostly there are different infection caused by the different types of microorganism. And the sample that is collected over here is a feces, and it is collected in a special container, which is a wide mouth bottle that can be tied scrutely once the specimen is taken up. And here the person is the patient is asked to give the feces voluntarily, and the collected feces is transferred to the laboratory for the further diagnosis. And here the type of method collection in the young children or the old patient is a short catheter that can be inserted to collect the feces. So here these patient cannot give the feces voluntarily. So in that case we can make use of a catheter to collect the specimen. And in case of parasitic infection, it is easy diagnosis because these parasites can be seen in the feces. Take for example the segments of tapeworm can be seen directly in the feces when observed in the microscope, and you can also go for the microscopic identification of the ova of parasites. And then threadworms usually lay these worms on the perianal area. So this can be collected with the help of a rectal swab or with the help of a sellotape mount, which can be put on the under the microscope and observed under the microscope. And finally, if the person is suffering from amoebic dysentery or suspected to be suffering from amoebic dysentery, you can go for the identification of free motile organism under microscope. So this is the way we collect the sample in the gastrointestinal tract, that is the sample is a feces. And coming to urinary tract specimens or urinary tract infections, as persons suspected to be suffering from urinary tract infections, the specimen of the choice is the urine. So here you can ask the person to give the urine voluntarily, and here the method that is used is called as a clean cache method. So in this particular method, the first dispersed urine is voided off, that is, it is eliminated, and midstream urine is usually taken into account. The reason for that is the first voided urine is containing different types of normal flora. So normal flora are nothing but the organisms that are normally present in our body as commensals. So these organisms are the one usually causes a confusion in the diagnosis. So to eliminate that particular problem, we eliminate the first voided urine. So only the midstream urine is usually collected in a container on which the different techniques are usually carried out. And if this this urine that has been collected takes time to reach the laboratory, then it should be immediately stored in a freezer at about 4 degrees centigrade, because by the time it reaches the laboratory, there is a chance that most of the organism will multiply and die due to their metabolic activities and concentration of the media. So if there is a time gap between the collection of the specimen and transport to the laboratory, you have to see that it has to be refrigerated around 4 degrees centigrade. And in case of children and patients, you can make use of different types of catheter to collect the specimen. And here, if the person is suspected to be suffering from mycobacterial tuberculosis, then the first voided urine, that is from the early morning urine, is said to be taken as a sample or a specimen, because it is more concentrated with the microbacterial species. So here the urinary tract specimen is a urine representing different types of urinary tract infections caused by mostly the gram-negative organism or the anaerobic organisms. Now coming to the genital swabs, mostly people suffering from the genitourinary infections caused by sexually transmitted diseases or the organisms. Here there are two most organism that usually causes the STIs, that is sexually transmitted diseases. So here the organisms are Neisseria gonorrhea, it can be Trypanosoma pallidum. So suspicious of Neisseria gonorrhea infection in females, the specimen that is collected is a cervical swab, or it can the swab that is usually dipped in an alginate or a cotton tipped applicator can be used to collect the specimen from the cervix of the female. And in male, it is a purulent discharge that can be directly seen under the microscope. And the patient, if it is asymptomatic, then cultures have to be carried out to identify the organism. So in the culture, Thayer-Martin medium is used to identify this Neisseria gonorrhea organism, and these grow. After the growth, they can be observed under the microscope as diplococci. So this is suspicious of Neisseria gonorrhea or gonococcal diseases. So the best method to collect the specimen is the swab. Now next, coming to the Trypanosoma pallidum infections, also the organism that causes sexually transmitted disease. Here the sample is directly taken from the lesion. So the organism causes characteristic lesions on the reproductive parts of the males or the females. So these lesions can be taken and observed under the microscopy. The organism cannot be cultured in the media because they are very sensitive to the environment. So you cannot grow them in the media. Directly you can observe them under the microscope, or you can identify this organism with the help of serological diagnosis. Now coming to the central nervous system diseases or the infection related to central nervous system, mostly these are systemic infections. So systemic infections are nothing but the infection that has completely spread throughout the body. There are certain organisms that usually causes this type of systemic infection. So in that case, if the brain or any important part of the body is affected, you can go for central nervous system specimen, that is a cerebrospinal fluid. And here the cerebrospinal fluid is usually collected in aseptic condition, and the procedure is called as a lumbar puncture, usually done by a needle, aseptic needle. And in case of viral infection, if suppose the person is suffering from viral infection that is systemic in nature, in that case you can go for cerebrospinal fluid or nose and throat swab for collecting the specimen. So here again you have to follow aseptic condition, because you should see to it that you are not introducing organism into the CSF. So complete sterile conditions and aseptic condition has to be maintained to take the specimen from the CNS or the cerebrospinal fluid. Blood stream specimen. Here the specimen of choice is the blood that is usually withdrawn from the vein, and here the vein is descended with the help of a tourniquet, and then the area from where the blood is collected is usually wiped with the help of soap and water, cleaned with an antiseptic, and then the needle, aseptic needle, is inserted and collected with the help of a syringe. The needle is discarded, and the content in the syringe is directly passed on to the container, which contains anticoagulants, and this container is used to carry out the other techniques. So here again you should see to it that you are not introducing organism into the human body with a contaminated needle. So all aseptic conditions have to be followed, and mostly the blood specimen are taken in case of onset of fever, after the onset of fever, because it represents the type of infection that is usually present in the body. And in case of intermittent fever, you can go for occasional drawing of the blood to identify the organism. Take for example in malarial patient. So for every 6 hours you can collect the blood to know at which stage that particular microorganism is usually present in our body. The next type of area which represents the disease area is the pleural or peritoneal cavities. These are said to be completely sterile in nature without any infection until unless there is any pyogenic or tuberculosis infection in our body. Again, the method of that is used in collection of cerebrospinal fluid is taken to collect the specimen from the pleural or the peritoneal cavity. Next is the abscesses, wounds or sinuses specimen. Here again the choice of specimen is a pus. So abscesses are nothing but the wounds. So due to the metabolic activity of the organism or the ulceration of that particular area, there is a possibility of discharge of pus in that area. So this pus can be collected with the help of a sterile container. No swabs can be used because that will lead to contamination. With the help of an aspiration or a piece or a wall of an abscess can be taken in a container. And this is about the different areas. And next coming to the eye, which is usually infected with most of the fungal organism. So here if there is a purulent discharge, you can collect it with the help of a swab, and if pus is not present, it can be usually scrapped with the help of a bacteriological loop that is made up of platinum. Here in case of bacteriological techniques, mostly the loop is made up of nichrome, but it causes the abrasion of the conjunctiva. So in order to overcome that, we are making use of a loop that is made up of a platinum to collect the specimen from the eye or the corners of the eye. And in case of chlamydia infection, you can go for chlamydial culture, that is take the specimen from the eyes and then immediately transfer it into the specific media which the chlamydia is usually used to grow, and transport it to the laboratory in the transport medium. So the transport medium is very important because there is a chance that this culture might be contaminated with the other organism. So in the case of transport media, we are trying to see to it that it is not allowing the activity of organism, neither the contamination with the other organism. Now in case of adenovirus infection, which is very less seen in case of eye infection, this the method of choice to collect is conjunctival swabs, or can also go for the throat swabs. And next is corneal ulcers. You can use a sterile spatula or a blade to collect a small amount of mass from that particular area to take it as a specimen. Now in case of endophthalmic infections, then you can go for aspiration from the vitreous by sterile syringe or needle. So there are different methods to collect the specimen from the eye. Now ear infections, usually it is in the external ear that you can see the infection, or sometimes it can be from the middle ear. So here again you can make use of swab or aspiration of the fluid from with the help of a needle to collect the specimen of ear infections. Now once you are done with the collection of the specimen, the next immediate thing that you do is you handle and then go for the processing of the specimen. So the work of the clinical microbiologist is in stepwise. First he looks for the part from which the specimen has to be collected. As already discussed, the results completely depends upon the quality of the specimen that you are going to collect. So first is the collection of the specimen, the mode of collection, how it is collected, then how it is transported to the laboratory where the further test can be carried out. So next step for the clinical microbiologist is to handle and then go for the processing of the specimen. So here in case of handling, once the specimen is collected, it has to be labeled correctly with the name of the patient, the type of test that is to be performed, and the type of different specific test or serological tests that are to be performed related to that particular organisms that has been usually collected. And then it has to be sent immediately to the laboratory or refrigerated in case if it is taking time to send it to the laboratory. And then you have to write a brief description or a form. A form should be filled pertaining to the type of tests that are usually performed on the specimen, and it has to be accompanied with the specimen to the main laboratory where the tests are usually carried out. In case of urgent report, then you can go for a special messenger to send to the laboratory to ask for the immediate report. So these are the handling procedures once the specimen is collected. Next is you go for the processing. The main part of clinical microbiology is to identify the organism from the specimen. So here the processing of the specimen should be done immediately without any delay. Otherwise, there is a chance of contamination, and then you have to follow the procedure from the first, that is collection of the specimen. So it should be done as possible, as immediate as possible to eliminate the risk of contamination. So here the tests that are done to identify the organism is the staining and the culturing. So processing, we have two major steps. One is the staining, to stain the organism as a smear and then observe it under the microscope for identify special characteristics of the organism based on morphology. And then go with the culturing to know the biochemical activities of the organism and ultimately identify a specific organism that is involved in the disease causation. So here this is called as diagnostic methods in clinical microbiology. So the first was diagnostic microbiology as a whole. So that is divided into collection of specimen, handling, and then methods that are involved. So there are different scientific methods that are involved to identify the organism. So specimen is collected, and then this can either be subjected to direct microscopic examination, after which it can be inoculated onto the culture media. So culture media is nothing but the media in which the organism is growing. So we can grow the organism in artificial laboratory, providing them with different conditions so that they can grow and we can identify the organism on a solid or a liquid culture. So the specimen from that is the blood or the bone marrow, once it reaches a clinical laboratory, the immediate step that we do is we inoculate onto the liquid media, which is a broth. Because to increase the number of microorganism or the desirable microorganism, it is first inoculated onto the broth. So here we have two different types of broth, one for the fastidious organism, the other one for the non-fastidious organism. The fastidious organisms are the one that require special nutrients for their growth, or they are exact in their nutritional requirement. So we enrich the media with special components such as blood, serum or egg, so that they allow the growth of fastidious microorganisms. So in the case of this organism from the blood, what we do is we go for the brain heart infusion broth for identifying the fastidious organism, and for non-fastidious organism it is the McConkey broth. So once a growth takes place for about 6 hours, 0.1 ml from each of the broth is transferred or subcultured on a blood agar, that is a solid agar. One that is from the brain heart infusion brought to the blood agar, and from McConkey brought to the McConkey agar. So here we can see the growth of the organism on a solid media. And the most important difference between the solid and the liquid media is you can see the organism as discrete colonies on the solid media, whereas in the liquid media it is a diffuse growth, so we cannot identify the growth of the organism. So it is inoculated onto the solid media to identify the growth of the organism. So this is incubated for about 37 degrees centigrade for 48 to 72 hours and seen for the growth of the organism. If there is no growth, then again the similar procedure, that is inoculating into the media with the 0.1 ml is carried out till about 2 weeks. So if there is no growth for 14 days also, the media can be discarded, and if there is a suspicious of any type of bacterial infection or bacteremia or septicemia, again you have to carry out from the first, that is from the patient you can collect the sample and grow them in the culture if there is any suspicious of bacteremia or septicemia. So this is how the processing of the blood samples usually takes place in the laboratory. So ultimately the growth of the organism can be seen on a blood agar or the McConkey agar, and you can identify the organism based on their morphology and the colony characteristics and the biochemical nature of the organism. Now, if there is a suspicious about a fungal infection, then there is a different media that is used. You cannot make use of the bacterial media to grow the fungi. So here we have a fungal media which is called as subrose dextrose broth. Subrose dextrose broth, first is a liquid media and then again inoculated into the solid media. But the time and condition in which we are incubating is different. So here we are incubating this fungal media for about 30 degrees centigrade, whereas for bacteria it is usually 37 to 40 degrees centigrade. For fungus we grow at 22 to 30 degrees centigrade for about 3 weeks. So immediately you cannot see the growth of fungal species on a media. It takes about 3 weeks to give the final report. So you never give the report within 1 or 2 days for a fungal identification. It takes about 3 weeks to grow on an artificial media. And for anaerobic bacteria, as there is a difference between aerobic bacteria and anaerobic bacteria, a special media is usually taken for the anaerobic bacteria, that is called as thioglycolate broth, which is again later on inoculated onto the blood agar. So, and it should be carried out in anaerobic jars, like providing anaerobic condition for the growth of anaerobic bacteria. Now, this is about the culture. The first thing what we do is we inoculate onto the culture and then go for the direct microscopic examination. Now, this microscopic examination is like utilizes different techniques. When we can go for wet films, hanging drop, and we can go for the stained smears to identify the morphology of the organism. So wet films can be done for the blood specimen to identify hemoparasites such as leishmania, you have trypanosoma and microfilaria. So hemoparasites are nothing but parasites that are present in the blood of representative of the systemic infection. And apart from that, you can go for gram staining, the basic staining technique to identify whether there is a gram positive infection or a gram negative infection. To enhance or to study in detail about the organism, you can go for fluorescent stains also to identify the organism's antigen related to the antibodies. Next thing is blood can also be contaminated or can be infected with the Plasmodium species. So for that sake, we can go for Leishman staining that helps to identify the Plasmodium involved in the disease malaria. And then you have the smear for bone marrow. The smear can be done to identify the fungal species, and the stain that is used is Giemsa stain. So for identify the organisms such as Histoplasma capsulatum. So different techniques like staining techniques you can go for wet films to identify the morphological characters of the organism by direct microscopic examination. Now next specimen we have with us is a cerebrospinal fluid that is collected from the CNS system, that is central nervous system, representative of systemic infection. Here as soon as we receive the cerebrospinal fluid, what we do is we subject the cerebrospinal fluid into two parts. One is centrifuge, the other one is uncentrifuged. So centrifuge part usually done in a centrifugation where the components get separated into sediment and a supernatant fluid, whereas the uncentrifuged pass can be directly taken for total count of WBCs, or mostly for the increase in the number of the WBCs indicates a pyogenic infections. So you can go for the total WBC count, or you can go for the collection of the CSF centrifugation into sediment and the supernatant fluid. The supernatant fluid is usually used to identify the microorganism antigens, or you can go for biochemical assays for their detection of proteins or nucleic acid or whatever it is. And then for this sediment part, you can go for the culture, that is you can inoculate either into the blood agar, you can inoculate into the McConkey agar. You have a chocolate agar, which is nothing but heated blood agar is a chocolate agar that helps to support hemophilus organisms. And you have Subroad agar for fungal species, and then you have Lowenstein-Jensen medium or Middlebrook medium for Mycobacterium tuberculosis. So this different cultures represent different types of bacteria. So in case of blood agar and McConkey agar, you can incubate at 37 degrees centigrade for 48 to 72 hours, and for SDA it is, as already discussed, it is 3 weeks, and for Mycobacteria it should be around 8 weeks at 30 degrees centigrade. So we should have a sound knowledge about the organism and the type of incubation we are keeping them. So for fungi it takes more time, whereas for Mycobacteria it takes the longer time for their growth onto the artificial media. And apart from that, you can go for a direct microscopic count where you can identify or see the organisms such as yeast cells involved in certain types of fungal infections, or acanthamoeba in cyst can be seen in the cerebrospinal fluid. So next is the conjunctival swabs that are usually taken from the conjunctiva and immediately inoculated onto the culture such as blood agar and McConkey agar and chocolate agar again for the hemophilus organism, and then you can inoculate onto the brain heart infusion broth for identification of certain types of bacteria which are very less in number. And then for anaerobic group of organisms, you can go for the anaerobic medium media, but hardly we can see any type of anaerobic organism related to the conjunctiva. So it is of no use or irrelevant for us to carry out anaerobic techniques in case of conjunctival swab. Then go for identification of organism that is associated with the eye, which is nothing but the gonococcal infection caused by Neisseria gonorrhea. So in that case, you have to inoculate them into a specific media which is called as a Thayer-Martin medium for identification of gonococcus, that is Neisseria gonorrhea. And if any suspicious of fungal infection, then inoculate onto the subroad agar. For the patient who are usually immunocompromised, that is in these particular patients there is an immunodeficiency disorders usually caused by HIV infection. So they completely like make down the immune system in this particular patient. So in that case they are called as immunocompromised patients. So they are more subjected to different types of fungal infections. So it is primarily the viral infection that makes the immunity comes down, so that the secondary infection can take place in this particular patient. So mostly the infections are from the fungus. So you can identify the organism in the subrods agar for the immunocompromised patient. So conjunctival swab, we have culture for bacteria, and then we have for Neisseria gonorrhea, and then finally for the fungal media in immunocompromised patients. Now the direct microscopic examination, here again you can go for gram staining for identifying gram positive or gram negative organism associated with them, and GMZ staining for fungi, and sometimes you can also see the inclusion bodies that are present in the organism with the help of this GMZ staining. And viruses can also be seen, that is there are some viruses that causes giant cells to develop. So with the help of different techniques you can identify the giant cells under the direct microscopic examination. Now coming to the corneal scraping, here the culture used is corneal ulcers. The segments are usually inoculated onto the media such as blood agar, McConkey agar, Saffrod's agar, BHIB or SDP, and usually for the anaerobic organism go for thioglycolate medium, and then blood agar, inoculate onto the blood agar. So anaerobic cultivation is different, carried out in anaerobic jars. So corneal scraping are thought to be infected with both like aerobic as well as anaerobic. If suspicious of anaerobic organism, go for the anaerobic media. And in the direct microscopic count, you can go for wet films, usually because they are fungal organisms that are usually associated with the corneal scraping. We can go for potassium hydroxide or lactophenol cotton blue staining technique to identify the fungi. And acanthamoeba cysts are also present that can be seen with the help of fungal staining, or gram staining can be done to identify the gram positive or gram negative organisms, and GMZ staining for fungus again, and conchoflor white. It is a special technique that is involving the fluorescent microscopy. So identifying the bacterial antigen with the specific antibody under the fluorescent microscope, the reagent that is used is conchoflor white. So this is about the corneal scraping. Now coming to the feces. Now coming to the feces, that is a specimen that the further tests are usually carried out for most of the organisms which are anaerobic in nature. So these organisms that are relative to this feces are usually the anaerobic group of microorganisms that are involved in gastrointestinal diseases, and these are called as Enterobacteriaceae members that are harbors the gastrointestinal tract of the human. So the specimen is a feces through which we are going to identify if there is any gastrointestinal infections in a particular human being. So here the culture that is used to identify organism is a McConkey agar to identify the lactose fermenting and non-lactose fermenting organism, and then we have deoxycholate agar, citrate agar for identification of Salmonella and Shigella. And apart from that, you can use thiosulfate citrate bile salt sucrose TCBS agar for identifying Vibrio involved in cholera. And then there is the growth of this Campylobacter organism. So here we have Campylobacter medium to grow this organism. Apart from the selenite, broth is also used, and here the organisms are grown and then subcultured on deoxycholate citrate agar and Campylobacter agar, and they are incubated for about 3 weeks as 37 degrees centigrade to identify Campylobacter or Salmonella or Shigella organism. And if there is any suspicious of fungal infection, go for the subroad agar. And for the toxin producing organisms such as Staphylococcus aureus, you can go for this blood agar or the mannitol salt agar. So feces can be subjected to different types of culture identifications. So mostly making use of anaerobic media to grow the anaerobic group of microorganism. Here we have another technique where we can identify the organism called a direct microscopic examination, where the feces has to be first concentrated. So and then wet films are made and identify the parasites with the help of Lugol's iodine solution. So identification of parasite is done for Lugol iodine, and then go for gram staining to identify pus cells or the bacterial cells or Vibrio or Campylobacter. Now we have intraocular fluids, pericardial, peritoneal, pleural and synovial cavity. So the type of subject that we are going to do to process the specimen is similar to that of the CSF, that is cerebrospinal fluid. That is, take the content, centrifuge it, half part centrifuge, half part uncentrifuge, and then supinate it, and the sediment can be inoculated onto the media. So in the case of intraocular, then it is mostly the fungal media, because the intraocular lesions usually contains the microorganism which are nothing but the fungi. So inoculate onto the subroads media, and the other medium that is used is a chocolate agar, and the microscopic technique that is used to identify organism is a Zeal-Nielsen staining. So most of the sinuses and pericardial or peritoneum cavities are usually infected with the mycobacterium, which causes a systemic infection in the later part of his cycle. So here you can identify the organism in this particular cavities with the help of direct microscopic examination by using Zeal-Nielsen staining. Now coming to the pus is collected from different parts of the area where there is a infection of the microorganism, and due to the ulceration of that particular area, pus is released. So this pus is usually indicative of anaerobic organism. So culture media that can be used is again blood agar, you have chocolate agar, you have BHIB, anaerobic culturing. We can make use of anaerobic culturing to identify anaerobic organisms, and then we have SDA for fungus, SDB again for fungus, Lowenstein, Jensen and Middlebrook medium for the identification of Mycobacterium species. And the direct microscopic examination, go for gram staining or Zeal-Nielsen staining to identify the mycobacteria. And the sputum specimen has to be processed before inoculating onto the media and identifying under the microscope. The sputum is first liquefied, and then it is concentrated in a centrifugation and decontaminated with alkali, and then inoculated onto the media. Media that is used is blood agar, McConkey agar, chocolate agar, subrods agar, and mostly the organism that is representative in sputum are the mycobacterial species that causes Mycobacterium tuberculosis. So a specific media for this organism is Lowenstein, Jensen media or Middlebrook media. And for identifying with the help of microscopy, go for the gram staining or Zeal-Nielsen or GMZ stain for the fungal staining. So mostly go for the the Zeal-Nielsen method to identify the Mycobacterium organism in case of patients suspected with Mycobacterium tuberculosis. So the best sample for identifying the Mycobacterium tuberculosis is the sputum. Next is the throat swab, so which is collected with the help of a sterile swab directly inoculated onto the media such as blood agar, potassium tellurite agar. So this potassium tellurite agar is mostly used to identify the diphtheria bacilli, that is Corynebacterium diphtheria, that is indicative of the pulmonary diseases in the children and in the patients. So special media is used over here for the throat swab to identify the Corynebacterium. Apart from that, you can go for SDA agar or anaerobic culturing for identifying anaerobic group of organisms. And the microscopic examination, go for gram staining, fluorochrome conjugated antibody technique, specially designed to identify the Corynebacterium diphtheria under fluorescent microscopy. Now coming to the urethral swab, again indicative of the urinary tract infection or the catheter collection of the urine. Culture media mostly used to grow organism of anaerobic type. So can go for blood agar, McConkey agar, chocolate agar, Thayer's Martin medium for Neisseria gonorrhea, and for SDA for the fungal species. Again, direct microscopic examination for gram staining, identify gram positive or gram negative. Mostly it is a gram negative organism that can be seen in the urethral swab or the urine culture. Now coming to the ear swab, mostly the pus is the specimen inoculated onto the SDA or CA or TMM, that is Thayer Martin's medium. Next is a urine sample inoculated onto the McConkey, and a special media that is called a cysteine lac electrolyte deficient medium. CLED medium is used to identify the organisms related to the urinary tract infections, and then we have blood agar, SD agar, and microscopy is usually by gram staining. Now this is all about the different culture techniques that we are using to identify or isolate the organism in a pure culture. Now pure culture is nothing but the culture containing a single type of organism derived from a single parent. So once we get the pure culture, it is easy for us to identify the organism and then tell the doctors like which type of infection it is, so that the doctor can treat the patient accurately with the certain types of antibiotics. So first thing that we have dealt in diagnostic microbiology is how we collect the specimens, what are the different ways and mode of collection of specimen. Next, how they are processed in the laboratory to isolate in pure form. And finally, it is final part of lab diagnosis or clinical microbiologist is to identify the organisms that have been grown on a pure culture and finally report to the doctors. So the last part in diagnostic microbiology is the identification of bacteria. So first it was the collection of the specimen, then the processing of the clinical sample by inoculating them onto the different media to isolate the organism in pure culture. And once the organism is isolated in pure culture, it is the final step to identify which type of organism is usually present, that is involved in the disease causation. So the last step is identification of the bacteria. Apart from that, you have other organisms also that causes the different types of infection. So for them, we have other techniques that can be used to identify the organism. But for identification of the bacteria, they can be identified based on the morphology, the corny characteristics or the cultural characteristics, and the other biochemical tests. So starting with the colony, that is a morphology, I will start with the shape, size, arrangement, motility, presence or absence of flagella, presence or absence of spores or the capsules. So based upon them you can identify the organism. So as discussed in the previous classes, each organism has a specific character with which it can be identified. Take for example the shape of the organism. Certain organisms are cocci in nature, certain organisms are bacilli in nature or rod shape. So based on the shape you can identify which type of organism it is. Now coming to the size, depending upon the size also you will know what type of organism is it, and the arrangement, how they are usually arranged under the microscope or in a media, you can know the organism. And then motility, so motility is usually by flagella, so depending upon the motility you can identify the organism. Not all organisms are motile in nature, so the organism which has flagella are motile. So presence or absence of flagella also helps us to identify the type of organism. And spores, especially the sporulating organisms are bacillus or the clostridium. So depending upon the presence of spores in an organism, you can identify the organism. And presence or absence of capsules, take for example diphtheria organism are capsulated, mostly involved in the infectious process. So these are the ways in which we can identify. The initial criteria for identification is the shape, size, arrangement of the microorganism and presence or absence of certain components in a particular bacteria. Next comes the staining reaction. So once we identify the organism under the microscope, then we go for the stains. So you can carry out a wet film or the hanging drop technique, or you can also go for the fixed stain smears to identify the organism under the microscope. So there are different staining techniques that have been developed to identify different types of organism. First, foremost thing to classify bacteria into two, that is gram positive and gram negative, is the gram staining developed by Christian's Grams. So you can classify the organism as gram positive or gram negative. Depending on that, you can identify the organism. So mostly gram negative organisms are anaerobic in nature and involved in the disease causation of gastrointestinal diseases. Take for example gram positive, they are usually associated with the respiratory disease. So this is how you can identify the organism by staining. Next can go for acid fast or non-acid fast staining, that is called as Ziehl-Neelsen staining. Acid fast organisms are usually the mycobacteria. So they can be identified with a special technique that is called Zeil-Nielsen method or acid-fast staining, and they cannot be stained with the gram staining. So this helps to identify the mycobacterium species. Next, to identify certain components in the organism, you can go for the staining of special appendages in the organism, such as the flagella stain. You have endospore staining. Presence or absence of endospores can be done or detected with the help of endospore staining, and you can go for capsular staining techniques. So these are various staining techniques by which you can identify the organism. So these are the preliminary tests that are usually done in identifying the bacteria, that is morphology and the staining. Later on, like if you want to get more information or detailed information of the organism, you can go for the cultural characteristics. So here the organisms are identified based on the growth on a solid media. So very less information is provided on a liquid media, whereas on a solid media, because this organism forms discrete colonies, you can identify the colony characteristics, and each organism has a differential colony characteristic that helps us in identification. So on a solid media, how they usually form colonies, with that we can identify the organisms. Take for example the shape of the colony. Some organism form circular, irregular or rhizoid in shape, that is the shape of the colonies on a solid media. Next is size. With the help of the scale, you can measure the size and you can know which type of organism it is, and depending upon the elevation, the size and the elevation of the colonies, that is whether it is an effuse, elevated, convex, concave, umbonated or umbilicate. Next, seeing the margins of the colonies. So here we have beveled or otherwise. So depending on that, also you can identify the organism's colony characteristics. And the surface of the colony, whether it is a smooth, wavy, or it is a rough, granular or papillate or glistening. And depending on the edges, whether it is entire, undulate, crenated or fibriated or curled. And color of the colonies. So for some of the organisms such as Pseudomonas usually form certain greenish coloration on the media. So with the help of that also you can identify it. And phototrophic bacteria also forms greenish coloration on the media. So that also helps us in identification. And for hemolysis also you can identify the organism, beta hemolysis, alpha hemolysis. So depending on that color colonies, you can identify the colony characteristic. Structure of the colony, that is whether it is an opaque, translucent or transparent. And coming to the consistency, how the consistency is, you can know whether it is membranous, friable or butyrous or viscid. So these are the colony characteristics that helps us to identify the organism, only seeing the colonies on the plate, but that is not the final identification. You have to go away further to identify the organism. Apart from that, you can go for resistance of the organism. Resistance is nothing but to tolerate a particular condition so that the organism is not killed. So there are certain organisms which can tolerate high temperature. They will not be killed if you increase the temperature. So resistant to heat, disinfectants, chemotherapeutic agents. So helps us to know the type of bacteria, whether it is a vegetative bacteria or a sporulating bacteria. So for a vegetative bacteria, you can easily kill at lower temperature, whereas the sporulating organism will usually require higher temperature to kill. So this helps us to get a sound knowledge about the resistance of the bacteria and helps us to identify the organism, whether it is a vegetative organism or a sporulating organism. Next is depending on their metabolism, whether they are aerobic or anaerobic, whether they require oxygen or not, whether they require carbon dioxide for their growth or not, and whether they usually produce certain pigments on a media or whether they causes the hemolysis of the RBC. So depending on that, you can know the metabolic activity of the organisms. Next is the biochemical test that we perform on the organism to identify the organism. So the biochemical tests are to identify the fermentations and other biochemical properties. So fermentation is a shortcut of respiration where most of the microorganisms or the anaerobic group of microorganism uses to produce energy. So in that course, they produces certain types of acids and alcohol that is beneficial for the human being. So identifying these characteristics of the organism helps us to identify the type of bacteria. So first I'll go with the sugar fermentation, also called as carbohydrate fermentation. So different organism has the ability to utilize different types of sugars. They can be monosaccharides, disaccharides or polysaccharides as their source of carbon. So So during that course, they usually produce certain types of acid. So they are usually, this test is carried out in test tubes where the carbohydrate is added along with an indicator. And in order to identify whether the organism is a gas producing or not, you insert a Durham's tubes in the inverted position. So depending upon the fermentation and utilization of carbohydrate, there is a change in the media. The media gets colorless if the carbohydrate is fermented. So this indicates that the organism has utilized the carbohydrate and produced the acid that has reduced the or decolorized the indicator that we have added. So production of the gas can be seen as the collection of the gas bubble in the inverted Durham's tube. So this helps us to identify the carbohydrate fermentation by different types of organisms. Next is the other biochemical test that are done is indole production. So indole production is usually done by gram-negative E. coli organisms. So when the organisms are growing in peptone water, they utilize tryptophan as a source of nutrition, and they produce an indole ring that can be identified by adding a reagent which is called as Kovacs reagent of 0.5 ml. So red coloring appears at the top, indicative the presence of E. coli organism. Next is the methyl red test, mostly used to see acid production. Most of the organism utilizes the sugar and produces acid, and brings down the acidity of the medium to around 4.5. So organisms are inoculated on media which is a glucose phosphate broth, and they are incubated for 5 days at 30 degrees centigrade, and to which to detect the production of acid we will add an MR reagent, that is methyl red indicator, that completely changes the color of the medium to red if the acid is produced. So positive is complete change in the medium to red color, and negative is yellow, remaining yellow in nature. So that is the MR positive organism. Next text is the Voges-Proskus test, that is used to identify the organism that produces acetyl methyl carbonyl from the pyruvic acid. It is an intermediate product that is formed by the organism, and this can be oxidized to diacetyl in the presence of alkali and atmospheric nitrogen, and the diacetyl reacts with the peptone of the medium, that is the glucose phosphate medium, ultimately changing the color to the red. So the reagent that is added is called as a Barrett's reagent, consisting of alpha naphthol and KOH. So when this is added, initially the color changes to pink in color in 2 to 5 minutes. So it deepens to around magenta and crimson in half an hour. When there is no change in color, that means it is negative. So positive is usually pink in color. That is a Voges-Proskus test. Next is the citrate utilization test. Some organism can utilize citrate as a sole source of carbon, and they can be detected by addition of indicator into the media. And here we have the change in the media, that is from green to blue, indicating a positive reaction. So these 4 tests, indole, methyl red, Voges-Proskus and citrate utilization test, are together called as IMVIC test to identify enterobacteriaceae members, usually the gram-negative bacteria involved in gastrointestinal diseases. So this is for enteric bacteria. Okay, so nitrate reduction test is usually carried out in a liquid media where 1% of KNO3 is added, and the reagent that is added to this to identify whether they are nitrate positive, that is they contain nitrate reductase as an enzyme or not, is sulfalinic acid, naphthylamine, and then we have acetic acid. So 0.1 ml of the reagent is added to see further test. So the ultimate positive test is in the form of red color. Next is production of ammonia. Ammonia, there are certain organisms who produces ammonia in the media that can be identified with the help of Nessler's reagent. And the brown is the positive indication of the production of ammonia, whereas yellow is a negative indication. Next, coming to the urease test, it is usually carried out in a special media called as Christensen's Jensen's media or Christensen's urease media. And the organisms which are positive that converts the urea to ammonia usually appears purple pink in color. So the result should not be given before 4 days. It is only after the 4 days that we have to give about the urease positive test. That is change in color to the purple pink. And then we have hydrogen production test. There are certain organisms that can decompose sulfur containing amino acid to produce the black or brown color change in the media. So sulfur containing amino acids are decomposed by special group of organisms, and this can be seen by the production of H2S. So the media contains less acetate medium, and the organism growing in that particular medium will ultimately form black or brown color change in the media. And then we have methylene blue reduction test. Here 1% of or 1 drop of 1% methylene blue is added to the media, and complete decolorization, if takes place, will indicate a positive reaction. It is because this methylene blue is an oxido reduction potential dye. It changes its color depending upon the oxidation or the reduction in a media. So when the organisms are continuously multiplying, the use of the media, and the indicator completely changes its color to decolorized, or it gets decolorized. So this is indicative of positive reaction. So methylene blue can also be used to identify the actively growing organisms. So catalase production is usually used to detect the organism that produces catalase, an enzyme that helps to kill the hydrogen peroxide that is added to water and oxygen. So when we add a drop of hydrogen peroxide to these organisms, if they are catalase positive, they show a brisk effervescence. Now coming to the oxidase reaction, it is used to identify whether the organism has this enzyme which is called as cytochrome oxidase, and oxidase reagent is mostly used. It is a tetraamine P phenylene diamine hydrochloride is a reagent that is used to identify whether the organism is oxidase positive or oxidase negative. So the organism which are oxidase positive usually ultimately forms black or brown color, and these are usually the positive organism with the oxidase reagent. Next method or biochemical test is the inoculation of the organism in an egg yolk. So organisms such as Clostridium usually produces an enzyme called as lecithinase that helps to develop the colonies, and this can be seen as a zone of clearance. So this is an indicator of the organism producing lecithinase as an enzyme. Apart from that, you can go for composite media. So it is very laborious for us to inoculate organism into different media and then see their characteristic. So nowadays we have got a composite media that has got all the characteristic, and on a single media you can identify different biochemical properties of the organism. Take for example TSI media. So here the organisms are inoculated on a single media, and different biochemical properties can be known on this particular media. It is called as triple sugar iron media. So on a single media you will know whether they are carbohydrate fermenting or not, whether they are gas producing or not, whether they are H2S producing or not. So single media is enough for us to know the different biochemical properties of an organism. So these are the classical techniques that we use to identify the organism in the media. So nowadays we have got more sophisticated methods to identify the organism, such as immunological methods, where we try to identify either the antigen or the antibody related to a particular organism. So that is nothing but the antigenic structure. So antigens are the organisms, are the components that are usually expressed on the organism, so which helps to produce the antibodies in the human body. So there is a high specificity between the antigen and the antibody. So this specificity helps us to develop various immunological assays to determine the specificity between antigen and antibody, and helps to ultimately diagnose a particular disease. So classical test takes about weeks together to identify the organism, but nowadays we have got immunological diagnostic techniques where within a minute or within seconds we can know whether there is an organisms associated with a particular disease or not. So this is an antigenic structure, that is immunological diagnosis. Next we can go for bacteriophage tapping. Very rarely used, because bacteriophage tapping helps us to know the intra-species in the organism. And then rapid identification methods are also used. They are called as molecular techniques. So you can use the DNAs and RNAs of the organism or the proteins of the organism to identify which type of organism is it. So the best method that we use in rapid identification method is the PCR techniques or gas liquid chromatographic techniques, and other molecular techniques such as plasmid fingerprinting, DNA fingerprinting, etc. So these are the different methods by which you can identify the organism. So in a diagnostic microbiology, we start with the collection of the specimen to final identification of the organism that helps us to cure the infection related to that particular infections.